This is part 3 of the chapter 20 lecture notes, and we left off on slide number 27, which is an illustration of how the human flu virus and the bird flu virus can interact within a host cell if they wind up in a host cell at the same time. And then the host cell can be tricked into combining them. So they're actually a new form of influenza virus. So we have a human influenza virus, which is an envelope virus. You can see the green capsid. You can see that the genome is RNA, but it is an envelope virus, and it is composed of N spikes, which are orange, and H spikes, which are pink. And just like any DNA or RNA, it can mutate when it replicates, and there's a mutation here. And then here we have several mutations that lead to a modified spike on the a modified H spike. Okay, so this is just showing you how the human influenza virus can mutate. And then when it mutates, that's the reason why we have to have a vaccine every single season, because it mutates so fast that the next season, the spike is different. So we have to have a different vaccine to account for that new spike. Okay. And those spikes, remember, are glycoproteins, which are proteins with carbohydrate chains attached to them. This shows you what can happen if the bird influenza virus and a human influenza virus end up in the same host cell. And that can happen a lot of times when people work in close association with birds, like people who work in chicken houses and turkey houses, and in very close association with birds. So anyway, these viruses can sort of combine, and when they do, the host cell creates a very, a new variety of virus, which is a combination of the bird and the human flu viruses. So that's another way that seasonal flu, human flu, and bird flu can, mutate and change. And every time it changes, it creates, a new struggle for us, a new challenge for us as humans. Okay, so emerging viruses. We hear about emerging viruses. These are new or previously uncommon illnesses. Coronavirus or COVID is an emerging virus, even though it's not brand new. Zika, Middle East Respiratory Syndrome or MERS, AIDS, West Nile encephalitis, Ebola hemorrhagic fever, and avian influenza or bird flu. These are, not all of them are brand new viruses, but they're new as far as human history goes. They're pretty new, okay? There are several types of events that can cause the emergence of viruses. The virus may extend its range. Example, the West Nile virus was transported to the United States and took hold in birds and mosquitoes. And a genetic mutation may occur. So these are the two main ways that emerging viruses become common. So the virus extending its range and genetic mutations occurring. This shows you an illustration of emerging viruses. These are in the 90s, and then we also have some 2014 and as high as 2019, which we know that one, of course, the SARS-CoV-2, which causes COVID-19. Okay, and it was named COVID-19 because it did begin in 2019. Here we are way over here. This is sort of the area where North Carolina is, and then we have Africa, and then we have the whole entire continent here that includes Asia and So way over here on the whole other side of the world, is where COVID-19 began. All right. We also in this chapter need to understand some not viruses, but they're similar to viruses in the fact that they are not living things, but they cause disease. So viruses are really best known for causing disease in plants and animals, herpes, HIV, cancer. But there are also particles called viroids and prions. Viroids are naked strands of RNA, and they typically affect crops. They cause crop diseases. So viroids, you need to make sure you know that they are just made of RNA. That's all they are, and they're associated with crop diseases. Prions are only composed of proteins. They're protein molecules with a contagious tertiary structure. If you remember from Bio 111, we learned that proteins had 4 levels of structure. They had a primary structure, which consists of a chain of amino acids. Secondary structure can either be a shape called a pleated sheet or helical structure. And the tertiary structure is the final 3-dimensional shape of the protein molecule. So when prions have a mutated tertiary structure that is contagious, and they cause TSEs, which are neurodegenerative diseases. And what they do is they destroy nerve tissue in the brain. Degenerative means they just get worse and worse over time and until they are fatal. They are untreatable and fatal. Mad cow disease is probably the most common, most well-known of those neurodegenerative diseases. Bovine spongiform encephalopathy, BSE, is mad cow disease in cows. And if humans eat brains, cow brains, basically, these prions can be transmitted to humans and cause the same disease. Now we're going to move on to section 20.2, which is only on prokaryotes, just so you can get kind of an overview of prokaryotes, which are going to include 2 domains, the bacteria and the archaea. So prokaryotes are single-celled organisms, and they do not have a nucleus, and they include organisms from the domains bacteria and archaea. So either one of those organisms, bacteria or archaea, are considered to be, or they are prokaryotes, okay? So we come back to Louis Pasteur, because Louis Pasteur showed us how to sterilize broth using a method called pasteurization. And his experiment basically looks like this. He showed that if you have a flask and you stopper it and you boil it, open it briefly and then close it back, 89% of those flasks will show growth of bacteria. And you can tell that there's growth in there because it will become cloudy. And that's what happens when bacteria grows inside of a broth, like a nutrient broth, kind of like chicken broth. And it looks and smells a lot like chicken broth. It actually smells a little worse. But anyway, 89% that are exposed to the air by opening, unstoppering the flask and then restoppering it show growth. And then if you do the same experiment, you boil the flask, you boil the broth to sterilize it, open it inside of a building, not outside of a building, but inside of a building, you see growth in 32% of those flasks. So what can you do to prevent the growth of bacteria? Well, you do the same experiment, but you use this opening here that has kind of an S shape. It kind of goes up and then down and then up again. So what happens is, even though the flask is open to the air, you boil the broth, kill all the bacteria. Even though the flask is open to the air, whatever comes in, the bacteria will fall and collect at that first little bend in the neck of the flask. It will not move, it will not travel up the flask. So the air at the top of that little pasteurization apparatus, the air at the top is pure. And so whenever you use this method called pasteurization, when you use a flask that has this kind of S-shaped bend in it, you can have 100% of flasks that show no growth. So this was how Pasteur developed his method of pasteurization, and it's how we pasteurize milk and a lot of our canned liquids now, canned and bottled liquids now, how we keep them free from bacteria and from making us sick. Now, prokaryotes, whether they are bacteria or archaea, their structure is very similar, even though there's differences, but we're going to talk about what's similar about them. First of all, they lack a nucleus. Bacteria and archaea both have no nucleus. They do contain a nucleoid region, and they do contain DNA in that nucleoid region, but they don't have an actual nucleus. They do have a cell wall, although their cell walls are not composed of the same exact chemicals, but they do both have cell walls. Some of them have flagella that help them to move. They do not have membranous organelles like mitochondria and endoplasmic reticulum and Golgi apparatus. They lack these. They may have plasmids, which are accessory rings of DNA that sometimes contain extra genes. Particularly in bacteria, the plasmids will often contain resistance genes, for example, for antibiotic resistance. The structure of all prokaryotes includes a cell envelope, which is made of a plasma membrane, a cell wall, and then a glycocalyx outside the cell wall. And that can be a solid capsule or kind of a slimy type of composition. And then inside of the cytoplasm of the prokaryotic cell is the nucleoid, which is the region that contains the DNA. And it's always DNA, it's always double-stranded DNA. It's just a ring of DNA instead of chromosomes that are the chromosomes like we have, There are ribosomes that make proteins, and in the case of cyanobacteria, which are the bacteria capable of photosynthesis, they can also have thylakoids, which we know are found inside of chloroplast. Even though the prokaryotic cell does not contain chloroplast, the cyanobacteria can have thylakoids, which help, enable it to carry out photosynthesis. And then some of them have appendages like flagella, conjugation pilus, and fimbriae. And they have serve different purposes. Here you can see the structure of a typical prokaryotic cell. We'll just look at it. Fimbriae are hair-like bristles that allow the cell to adhere to surfaces. The nucleoid is the region that contains the long circular bacterial chromosome. A conjugation pilus allows the bacteria to transfer DNA to another bacterial cell. Flagellum will allow it to swim, to move. A capsule is composed, it is the part we call the glycocalyx, composed of a polysaccharide layer. The plasma membrane is the innermost layer. So you have the plasma membrane that comes first. It is composed of a phospholipid bilayer. And then you have the cell wall. In bacteria, this is composed of peptidoglycan, but it's different in archaea. Then you have ribosomes